Now we are talking about the carbohydrate metabolism. We are going to discuss first the glucose metabolism. In the glucose metabolism, we will discuss the breakdown of the glucose, that is called as glycolysis, and then we will discuss the synthesis of the glucose, that is called as gluconeogenesis. Then we will talk about the glycogen metabolism. The glycogen synthesis will be called as the glycogenesis, and the breakdown of the glycogen will be called as glycogenolysis. Then the galactose metabolism and the fructose metabolism. These are the major metabolism that we will discuss. Then we have the minor metabolism pathways such as the HMP Shunt uronic acid pathway. We will discuss afterwards. Starting with the first one, that is the glucose metabolism. Now in the glucose, first we are going to discuss the glycolysis. Glycolysis is also called as the AMP pathway, that is embedded Mayer-Half-Parnas pathway, where we are going to break the glucose by 10 multiple different steps. The glucose, which is a 6 carbon molecule, via these 10 steps ultimately will convert into pyruvate, which is having 3 carbon. And because pyruvate is having 3 carbon, so we are going to get 2 molecules of pyruvate, plus we are going to get a certain amount of energy. Out of these total 10 steps, 7 steps are going to be reversible and 3 steps are going to be irreversible. So let's see how these steps are going to happen. We start with the glucose molecule, and then we are going to do the first reaction, that is the activation of the glucose by giving the energy, and where we will bring the ATP near to this. So the phosphate will be transferred on the 6th carbon of the glucose. We are going to generate the glucose 6-phosphate. This is going to be an irreversible process, and whenever we transfer the phosphate and the ATP is coming in the picture, the name of the enzyme is going to be kinase. So we can use the hexokinase or glucokinase. Hexokinase is a non-specific enzyme that can work on any hexose. Glucokinase is specific to glucose. Then the further reaction will happen. Then we will reach to the third reaction, that is going to be an irreversible reaction, that is the phosphofructokinase 1, and that is going to be the most lazy enzyme of the pathway, means it is called as the rate limiting step. Then we are going to do some further reactions, and ultimately in the fourth reaction, the fructose 1,6 biphosphate will split into 2 small molecules, that is the 2 almost equal part. The fructose 1,6 biphosphate, it was having 6 carbons and 2 phosphates. So we are going to divide 3 carbon with 1 phosphate, 3 carbon with 1 phosphate. This is how we are going to divide. The 3 carbon molecule that you are going to get is glyceraldehyde 3-phosphate, and the other one you are going to get is dihydroxyacetone phosphate. The dihydroxyacetone phosphate will ultimately convert into glyceraldehyde 3-phosphate. So after the 5 reactions, actually we start with the glucose molecule, and when we complete the 5 reaction, we are having the glyceraldehyde 3-phosphate, and this side also we are having the glyceraldehyde 3-phosphate. Then whatever is the next 5 reaction that is going to happen with this side of the glyceraldehyde 3-phosphate, the same is going to happen with this side also, and ultimately we are going to generate the pyruvate. So we are going to generate 2 molecules of pyruvate. This glucose will ultimately convert into 2 molecules of pyruvate. The last step is also going to be irreversible. Now, what are the points that we must remember out of the glycolysis? See, there are several toxins which can inhibit the glycolysis, and one of the very important toxin is sodium fluoride. The sodium fluoride inhibits the enzyme enolase. Now, why it is important? Because we use it during the sample collection. When we want to do the sample collection, say for example, you want to do a glucose estimation, what you are going to do is, in the vial where you are going to collect the blood, you are going to put the sodium fluoride in that. So what is going to happen? The glycolysis will not happen. And glycolysis is a pathway that occurs in the cytoplasm, means can occur in the RBCs also. So if I collect the blood for the glucose estimation right now, and if I will estimate after 2 hours, during that period the RBCs can consume the glucose and I may get a false low result. So what we will do, we will add the sodium fluoride, so the glycolysis will not happen by inhibiting the enolase enzyme. Then there are some other toxins. Iodoacetate can inhibit the glyceraldehyde 3-phosphate dehydrogenase, and arsenate can inhibit the phosphoglycerate kinase. But they are not clinically used, so they are not much asked. What is much asked is going to be the sodium fluoride, which is going to be inhibiting the enolase. What are the other things to be remembered? The rate limiting enzyme of glycolysis, the phosphofructokinase. There are total 3 irreversible steps are going to be there. How we remember? The first one, the last one, and the rate limiting one. The first, the last, and the rate limiting one. The first one is hexokinase, the last one is pyruvate kinase, and the rate limiting one is PFK1, phosphofructokinase 1. When it comes to energetics, in this entire process, we are going to invest 2 ATPs. In the first 5 reactions, out of the 5 reactions, actually it's the first one and the third one, we invest 1 ATP. So total investment is 2 ATPs. And what we are going to get in return is the 9 ATPs. So out of 2 ATPs, we are going to get 9 ATPs. So the net benefit from the glycolysis is going to be 7 ATPs. When it comes to the substrate level phosphorylation, means the reactions where you are going to generate the energy directly in the form of ATPs. In the glycolysis, we are going to do the substrate level phosphorylation 2 times, the phosphoglycerate kinase and the pyruvate kinase. These are the 2 reactions where you are directly going to generate the ATP. When it comes to the regulation of glycolysis, see what we are doing in the glycolysis is we are breaking down the glucose and ultimately we are making the pyruvate. So basically we are breaking the glucose molecule. And who loves to break the glucose? Yes, it is insulin. Insulin wants to decrease the glucose level. How insulin will decrease? Insulin will stimulate the glycolytic enzyme. If we will stimulate the glycolytic enzyme, the glucose will convert into pyruvate. So insulin is the one that is the stimulator of glycolysis. And the opposite of insulin, who is the opposite hormone of the insulin? Glucagon. Glucagon is going to inhibit this because glucagon always wants to save the glucose. So these are the one stimulator and inhibitor. What are the other things? We already know about the negative feedback. See, what is the meaning of negative feedback? If the products are more, what are the products of glycolysis? Pyruvate, ATP, these are the products. If they are more, they are going to inhibit this process. They will tell the enzymes that no need to do the reaction, we are already there. So in any cell, if the pyruvate, if the ATPs, they are more, they are going to give the negative feedback. They are going to inhibit. Whereas if the substrate is more, if the glucose or the ADPs are more, they are going to stimulate this process. So substrate, they are going to be stimulator, products are going to be an inhibitor. What we have understood so far is that from glucose we can convert into pyruvate and we are going to generate 7 ATP. But what is going to happen with this pyruvate molecule afterwards? See, this pyruvate, it will depend that the cell is containing the oxygen or not containing the oxygen. If we go towards the anaerobic pathway, if the oxygen is not there, the pyruvate will convert into lactic acid. And to convert the pyruvatic acid into lactic acid, we will consume 2 NADH. 1 NADH is equal to 2.5 ATPs. So basically you are going to consume 5 ATPs. What was your net benefit till this point of glycolysis? 7 ATP. But if you do this reaction, you are going to consume 5 ATPs. So it is going to be 7 minus 5 comes out to be 2 ATP. So if they ask that anaerobic glycolysis will give you how much ATP? Only 2 ATPs. But if the oxygen is available, which is going to be there in the mitochondria, then this pyruvate will enter inside the mitochondria and the pyruvate will convert into acetyl-CoA. The pyruvate will convert into acetyl-CoA. And that acetyl-CoA is going to run the Krebs cycle. So basically glucose till pyruvate, it is called as the glycolysis. Glucose till pyruvate is glycolysis and from acetyl-CoA onwards, it is called as the Krebs cycle. So I can say that this particular reaction where the glucose is converting into acetyl-CoA, this is a bridge reaction or a link reaction between the glycolysis and the Krebs cycle. This is a bridge between the glycolysis and the Krebs cycle. So we can say like this, this reaction is very important to connect the glycolysis from the Krebs cycle. Now, if you talk about the cancer cells, they consume the glucose at a very rapid speed. Although they contains the mitochondria, they contains the oxygen, but they deliberately will do the anaerobic glycolysis. So they consume a lot of glucose actually. So this is called as the Warburg hypothesis. So a question is asked that in the cancer cell, which type of glycolysis we are normally seeing? It is the anaerobic glycolysis. This is called as the Warburg hypothesis. Let's understand one more extra point, and I want the answer from your side this time. See, the glucose converts into fructose 6-phosphate, the PFK1 will do the reaction and ultimately we generate the pyruvate. This is the normal glycolysis. But there is one more brother of the PFK1 is there, that is called as the PFK2. If the PFK2 works, then the fructose 6-phosphate, which was supposed to convert into 1,6-phosphate, now it converts into 2,6-biphosphate. Now, the point is this fructose 2,6-biphosphate, it is going to stimulate this process. So sometimes they ask that, what is the role of fructose 2,6-biphosphate? Is it stimulating the glycolysis or inhibiting? It is going to stimulate. Why it is stimulating is because the moment it will be formed, see, any product once it is formed, it does not want to be the reaction to occur more. So once the fructose 6-phosphate will convert into fructose 2,6-biphosphate, it is going to inhibit its own enzyme. It will tell that no need to do this reaction and it will stimulate the alternate pathway so that this fructose 6-phosphate will convert towards this side. So fructose 2,6-biphosphate is a stimulator of glycolysis. There is something called as the Rappaport-Lubring shunt, also called as the 2,3-BPG shunt. You must have heard in this thing in the physiology. The RBC helps in the transport of oxygen. How the RBC do the transport of the oxygen? The glucose, see, normally the glycolysis when we do, one of the intermediate that we generate is 1,3-biphosphogylicate. And that 1,3-biphosphogylicate converts into 3-phosphoglycerate. And during that process, we generate 2 ATPs. But sometimes in the RBC, the 1,3-biphosphogylicate does not convert into 3 directly. First, it converts into 2,3-biphosphogylicate, and then it will convert into 3-phosphoglycerate. So the 2 ATPs that we were supposed to get, we are not going to get now, if you are doing the Rappaport-Liebring shunt or the 2,3-BPG shunt. So the RBC, sometime they will do like this. And if they will do like this, they are going not to generate these 2 ATPs. But why they are doing this? What is the requirement of this 2,3-BPG? See, these 2,3-BPGs in the RBC, it helps in the loading and the unloading of the oxygen molecules. Because the function of the RBC is to transport the oxygen. How the RBCs transport the oxygen? They generate this molecule that helps in the loading of oxygen as well as the unloading of the oxygen. So if they ask that in the Rappaport-Liebring shunt, what is the net energy gain? The net energy gain is going to be 0 ATPs. So there are 3-4 questions that we have understood from the glycolysis. If they say glycolysis gives how much ATP and nothing is specified, then the answer is going to be 7 ATPs. But if they say anaerobic glycolysis, if they specifically use the word anaerobic glycolysis, then the answer is going to be 2 ATPs. And if they say Rappaport-Liebring shunt, then the answer is going to be 0 ATPs. 0 ATPs. And if they say glycolysis in the RBC, that gives how much ATPs? See, in the RBC, we don't have mitochondria, so we have to do the anaerobic glycolysis. So RBC glycolysis can only give the 2 ATPs, and if you do the Rappaport, those 2 ATPs will also not be getting, so we will get the 0 ATPs. So RBC can get 2 ATPs or can get 0 ATP, depending that RBC is doing the Rappaport or not doing the Rappaport. The next is the link reaction. The link reaction is going to link the glycolysis from the Krebs cycle. So it is a bridge between the glycolysis and the Krebs cycle. It is going to occur in the mitochondria, where the 3-carbon pyruvate will convert into 2-carbon acetyl-CoA. And in this entire process, the NAD will convert into NADH. We are going to remove 1 carbon dioxide because 3 to 2, and we are going to insert a coenzyme A. So actually in this process, there are 3 reactions are happening. You are removing the carbon dioxide, you are giving the coenzyme A, and you are converting the NAD into NADH. So to do this reaction, we require the enzyme that is called as the pyruvate dehydrogenase complex. It is going to be a complex enzyme and it will have 3 subunits, E1, E2, E3. E1 is going to do the decarboxylation, E2 will do the transacetylation, and E3 will do the dehydrogenase reaction. So you should know the name. E1 is decarboxylase. The other name of E1 is pyruvate dehydrogenase, E2 is transacetylase dihydrolipoyl, and E3 is the dehydrogenase dihydrolipoyl. And what are the coenzymes? E1, the coenzyme is B1. This is how we have remembered E1, B1. E3, what is the coenzyme for E3? B3 and B2. E1 is B1, E3 is B3, B2, and E2 is B5, that is 25. This is how we have remembered 25. Now, this is what we have understood so far about the glycolysis. The glucose can convert into pyruvate. The pyruvate either converts into lactic acid or can convert into acetyl-CoA via the pyruvate dehydrogenase complex. There are some unfortunate children are there and they don't have this enzyme. Just imagine that if this enzyme is not there, pyruvate dehydrogenase, if this enzyme is not there by birth, the reaction will not happen and the pyruvate will be forced to go towards where? Lactic acid. There will be more lactic acid production. The child may develop lactic acidosis. So this is called as the congenital lactic acidosis. That is due to deficiency of what? Pyruvate dehydrogenase complex. Plus, there is one more way by which this process can happen. See, in the pyruvate dehydrogenase complex, we have 3 subunits, E1, E2, E3. What is the coenzyme for E1? B1. So if by any chance there is a vitamin B1 deficiency, the E1 will not work, the enzyme will not work, and again the pyruvate will more convert into lactic acid. So vitamin B1 deficiency can also lead to lactic acidosis. The next we are discussing is the Krebs cycle or the TCA cycle. TCA is because it is also called as the tricarboxylic acid cycle. This is going to occur in the mitochondria. So all the enzymes of the Krebs cycle should be there in the mitochondrial matrix. But there is one enzyme which is not found in the mitochondrial matrix. Instead, it is found on the inner mitochondrial membrane. There is one enzyme of the Krebs cycle which is found on the inner mitochondrial membrane, and the name of that enzyme is the succinate dehydrogenase. Succinate dehydrogenase, it is found on the inner mitochondrial membrane. Who is going to start the Krebs cycle? It is the acetyl-CoA. So the acetyl-CoA molecule will come and will start the Krebs cycle. It will do all these 8 reactions and ultimately it is going to convert into oxaloacetate. Now this oxaloacetate molecule again will unite with the next acetyl-CoA which is coming and will make the citrate and will again do the reaction. In this entire process, you are going to generate 3 molecules of NADH, 1, 2 and 3. There are 3 NADH will be generated. We are also going to generate 1 FADH and 1 ATP. So the total energy that we are going to generate, 3 NADH, 1 FADH and 1 ATP. This is what we are going to generate in the Krebs cycle. Where it is going to occur? It is going to occur in the mitochondrial matrix. What are the points to be remembered? See how we have remembered the sequence of the intermediate. We have used one mnemonic. The mnemonic was like this. Citrate is Krebs starting substrate for making oxaloacetate. Citrate is Krebs starting substrate for making oxaloacetate. So this is how we are going to remember the various intermediate. What are the other points that you must remember from the Krebs cycle? See, the first is the inhibitor, and among the inhibitor, the most important inhibitor is malonate. Malonate inhibits the succinate dehydrogenase. The succinate dehydrogenase enzyme is inhibited by a malonate. Then we have some other inhibitors such as the fluoroacetate, which is going to inhibit the aconitase, and arsenite can inhibit the alpha ketoglutarate dehydrogenase. Total how much ATPs we are going to get? I told you 3 NADH, 1 FADH, 1 ATP. If you convert everything into ATP, it comes out to be 10 ATPs. We are going to get 10 ATPs. Then what is the when is the substrate level phosphorylation is happening? Substrate level phosphorylation means the formation of the ATP at the time of reaction. In which reaction you are generating the ATP directly at the time of reaction? Only one time in the Krebs cycle, and in that the name of the enzyme is succinyl thiokinase. Succinyl thiokinase, and what is the substrate? Succinyl-CoA. What is the starting substrate of the Krebs cycle? Acetyl-CoA. The starting substrate is going to be acetyl-CoA. The succinate dehydrogenase, the coenzyme, or sorry, the cofactor for succinate dehydrogenase, it is iron. Iron. Just before the Krebs cycle, we have discussed about the link reaction. In the link reaction, the name of the enzyme was pyruvate dehydrogenase complex. Do you remember there were 3 subunits, E1, E2, E3? What is the coenzyme of E1? B1, E2, B5, E3, B3 and B2. In the Krebs cycle also we have one enzyme which have the similar coenzymes. The name of that is alpha ketoglutarate dehydrogenase. It also have the same coenzymes. So they ask a question that which enzyme of the Krebs cycle it is structurally similar to the PDH complex. The answer is going to be alpha ketoglutarate dehydrogenase. It is very much structurally similar to the PDH complex. So if we have one glucose molecule, and I do all the oxidation reaction with all the possible enzymes, how much ATP we can get? See, first we'll do the glycolysis, that is going to give us 7 ATPs. Then we will do the link reaction, the pyruvate to acetyl-CoA, that is going to generate the NADH molecule. And since we have got 2 molecules of pyruvate, so the link reaction is going to occur for 2 times. We are going to generate 2 NADH. 1 NADH is 2.5 ATP. So link reaction will give us 5 ATPs, and ultimately we are going to get 2 molecules of acetyl-CoA that is going to run the Krebs cycle for 2 times. Once time Krebs cycle will give 10 ATP, 2 times will be 20 ATPs. So the net energy gain 7 plus 5 plus 20. From one glucose molecule, maximum how much ATPs we can get? We can get 32 ATPs. The maximum ATPs that we can get is 32 ATPs. Okay. The next process that we are discussing is the gluconeogenesis. Gluconeogenesis, as the name is indicating, genesis means we are going to synthesize, and what we are going to synthesize? The glucose we are going to synthesize. From which substrate we are going to synthesize? The non-carbohydrate substrate, means such as amino acids, fatty acids, pyruvate. From that we are going to make the glucose, that is called as gluconeogenesis. So they ask 2 questions from the substrate. What is the major substrate? The major substrate is amino acid, and in the amino acid also, which amino acid is the most glucogenic? Alanine. And the second question they ask is, what is never a substrate of gluconeogenesis? You can never synthesize glucose from which molecule? Acetyl-CoA. Acetyl-CoA is never a substrate of gluconeogenesis. Okay. Now we are going to see that how we are going to make the glucose from these various substrate. First from the pyruvate. How we are going to generate the glucose from pyruvate? Can you tell me that in the glycolysis, total how many irreversible steps we had? 3. First, last, and the rate limiting one. First, glucokinase, rate limiting one, PFK1, and the last was pyruvate kinase. Now, right now, the task is we are standing here at pyruvate, and I want to go back all the way till glucose. So, I will be having these 3 hurdles. So, I have to bypass them. First, this pyruvate will enter in the mitochondria, and the pyruvate will convert into oxaloacetate. Oxaloacetate, 3 carbon to 4 carbon. So, the name of enzyme is carboxylase. Pyruvate carboxylase. Then this oxaloacetate will come out with the help of malate shuttle back in the cytoplasm, and this oxaloacetate then will convert into phosphoenol pyruvate. So actually we have bypassed one hurdle. We went inside, we came via the oxaloacetate, and we have bypassed one hurdle. Now we are going to use the same enzymes because they are reversible. We'll come here and we'll stuck here. Now we are going to use some alternate enzyme, that is to convert the fructose 1,6 biphosphate into fructose 6 phosphate. We will use the fructose 1,6 biphosphatase and we will reach here. Then we are stuck here. We will use the alternate enzyme to convert the glucose 6 phosphate into glucose 6 glucose. We are going to use the glucose 6 phosphatase. So to bypass these 3 hurdles, actually we are using 4 alternate enzymes and 1 shuttle. And ultimately from pyruvate, we will be able to make the glucose. Now coming to the next one, that how we can convert the lactate into glucose. See, what happens is when the muscles are consuming the glucose molecules, they generate the pyruvate, and if you are doing exercise, the muscles will consume the glucose very fast. The muscle will do anaerobic glycolysis, and the product is going to be lactic acid. And that is why you have seen that when you are doing the gym, after the gym, your muscles are looked very ripped. They looked swollen because the lactic acid accumulated there. But after some time, slowly this lactic acid will come out, and this lactic acid will go towards the liver. What the liver will do with the lactic acid? Liver will convert the lactic acid into pyruvate, and we know that from pyruvate we can make the glucose. And then this glucose can come back via the blood to the muscles for the energy purpose. So actually, we were supposed to discuss only this much part. This is the gluconeogenesis from the lactic acid, but entire cycle is actually called as the Cori cycle. This is called as the Cori cycle. Then the next is how we are going to convert the various amino acid into glucose. The most important amino acid from which we make the glucose is alanine. Alanine can convert into pyruvate via the transamination reaction. If you remember from the chapter of amino acid, SGOT, SGPT, yes, that was the transamination reaction. So alanine can convert into pyruvate. Alanine can convert into pyruvate. We have used one mnemonic also, if you remember, SOLP. SOLP, L stands for alanine. Alanine can convert into pyruvate. And that is the transamination. And once you make the pyruvate, pyruvate can convert into oxaloacetate, and from oxaloacetate we can make the glucose. And this is how we can make the different amino acid into glucose. We can convert phenylalanine, tyrosine, aspartate, valine, isoleucine. We can convert all these various amino acid into glucose. But the most important one is the alanine, the major substrate, right? So this is how we can make the glucose from different substrate. Even we can make the glucose from the fatty acids, particularly the oat chain fatty acid. When we are going to do the beta oxidation of the oat chain fatty acid, the last product we are going to get is a 3-carbon molecule, propenyl-CoA. This propenyl-CoA can convert into methyl-malonyl-CoA, 3 carbon converting into 4 carbon, so the name of enzyme is carboxylase. This methyl-malonyl-CoA can convert into succinyl-CoA, and then the succinyl-CoA can convert into oxaloacetate, and we know that from oxaloacetate how to make the glucose. So from fatty acids, particularly the oat chain fatty acid, we can make glucose. What is the important MCQ from here? See, if you see the reaction when the methyl-malonyl converts into succinyl-CoA, the name of the enzyme is methyl-malonyl-CoA mutase, and the coenzyme is vitamin B12. Means this reaction only occurs if the vitamin B12 is active. So if I want to see the intracellular activity of vitamin B12, I will see that how good this reaction is happening. Say for example, there is a person in whom the vitamin B12 is not working properly, then this reaction will not happen. And if this reaction will not happen, what is going to accumulate in the body? Methyl-malonyl-CoA. So in any MCQ, if they say methyl-malonyl-CoA is more, means what they want to indicate that there is a vitamin B12 deficiency is there, right? Now coming to the regulation of gluconeogenesis. When it comes to regulation, what we are doing? From the non-carbohydrate substrate, we will make the glucose. Does insulin wants to make glucose? No, insulin never supports such reactions where the glucose is a product. So insulin is going to be a strong inhibitor of gluconeogenesis. Who wants to make glucose? Glucagon. Glucagon is going to be a stimulator. We know that acetyl-CoA is not a substrate of gluconeogenesis. It is not a substrate, but yes, it can stimulate the gluconeogenesis. Acetyl-CoA is not a substrate, but it can stimulate the gluconeogenesis. The next that we are discussing is the glycogen metabolism. First, we need to understand what is this glycogen is. See, when you are eating extra glucose molecule, when we are consuming the food, there is so much of glucose is there, all the glucose molecule will absorb and this will go to the liver. Now, the liver is not able to understand that so much extra glucose is coming, what to do with this? So the liver thought that let's store the glucose molecule. So as and when required, when the patient will be in the fasting state, we will cut the glycogen molecule and we'll get the glucose back. So these glucose molecules will be clubbed together and a huge molecule will be formed called as glycogen. So glycogen is actually a homopolysaccharide of glucose. And the glycogen will be stored in the liver as well as it is going to be stored in the muscles. Because muscles are the one that requires a lot of energy. So what the liver will do, liver will give him the glycogen. You keep the glycogen with you as and when you want, you cut it and you eat it. So the energy purpose will be solved for the muscle. But how to make this glycogen? See, to make the glycogen, what we have to do is we have to make the glucose molecule sticky so that they can club together. In the liver, there is a protein called as glycogenin. The glycogenin is a protein. On that molecule, we are going to attach the sticky glucose molecules. The sticky glucose molecules are called as the UDP-glucose. So what the liver will do, whatever extra glucose that is coming, first convert that glucose into UDP-glucose. They are very sticky. So they will attach together. These UDP-glucose molecules, they are going to attach together. First, we are going to attach the glucose molecule in the linear sequence. And in the linear sequence, the direction of the bond is going to be alpha 1,4. At the same time, we can make the branching points also, and we can make the branches. To make the branching point, the direction of the bond is the first carbon unites with the sixth carbon, that is alpha 1,6. Then again, it is going to be a linear sequence, and in the linear sequence, again, it is going to be alpha 1,4. So in the glycogen, if they ask, what is the direction of the bond between the glucose molecule? In the linear sequence, it is going to be alpha 1,4, and only at the branching point, it is going to be alpha 1,6. How the glycogen metabolism occurs? See how the glycogen synthesis occurs. Glycogen synthesis is called as glycogenesis. It is going to occur when you are in well-fed state. When we have a lot of glucose in our body, we are going to make the glycogen. And how we are going to break down the glycogen? That is called as the glycogenolysis. That is going to happen in the fasting state. Let's see the process. First, we are going to see the glycogenesis. When we are in the well-fed state, the excess glucose will convert into glucose 6-phosphate. The glucose 6-phosphate will convert into glucose 1-phosphate, and that will convert into UDP-glucose, the sticky glucose molecules. And now, with the help of the enzyme glycogen synthase, first we are going to make a linear sequence. And with the help of branching enzyme, we are going to make the branches. And ultimately, what we are going to make? Glycogen. That will be stored where? In the liver and the muscle. This is how the glycogen synthesis occurs. The rate limiting enzyme in the glycogen synthesis, it is going to be glycogen synthase. Glycogen synthase is going to be the rate limiting step. When we are in the fasting state, we are going to cut the glycogen. So in the fasting state, we will cut the glycogen. To cut the glycogen, the main important enzyme is glycogen phosphorylase. This is the rate limiting enzyme, and the coenzyme is vitamin B6. When you cut the glycogen molecule, we are going to get a molecule that is called as the glucose 1-phosphate. We don't get the UDP-glucose, we are going to get glucose 1-phosphate. The glucose 1-phosphate then can convert into 6-phosphate and then ultimately can convert into glucose. To convert the glucose 6-phosphate into glucose, we require a special enzyme that is called as glucose 6-phosphatase, which is only found in the liver. Now here is a tricky question, that glycogenolysis means the breakdown of the glycogen. Actually, it is occurring in two organs, liver and muscle. Both are cutting the glycogen. Both will cut the glycogen in the same manner till this point, till the glucose 6-phosphate. But the last enzyme is only found in the liver. So the liver is the only organ which can convert the glucose 6-phosphate into glucose. The muscle can't do that. So if they ask what is the last product of glycogenolysis in the muscles, in the muscles, the product is going to be glucose 6-phosphate. And in the liver, what is the last product? Glucose. Because liver has which enzyme? Glucose 6-phosphatase. Plus, they asked one question, that This is glycogenesis. This is glycogenolysis. Can you tell me one enzyme which is common in both the pathway? There is only one enzyme which is common in both the pathway, and the name of that enzyme is phosphoglucomutase. Phosphoglucomutase, which is common in both the pathway. There are so many enzymes which are involved in the glycogen metabolism. By any chance, if any enzyme is not there, the intermediate of the glycogen metabolism will accumulate in the body. And these diseases are called as the glycogen storage diseases. It's a very hot favorite topic of the exams. If the glycogen metabolism is not happening properly, any enzyme is deficit, the glycogen will accumulate in the body. So what are the various glycogen storage disease that we have? These are the various glycogen storage disease. The glycogen storage disease type 0, that is deficiency of glycogen synthase. If the glycogen synthase is not there, we can't make the glycogen. Then we have the type 1, 2, 3, 4, 5, 6 likewise. The mnemonic that we are going to use is ABCD. If they say Anderson disease, it is due to deficiency of branching enzyme. Anderson disease, it is due to deficiency of branching enzyme. Cori disease, it is due to deficiency of debranching enzyme. Cori is due to debranching, ABCD. McArdle, it's due to deficiency of muscle glycogen phosphorylase. Hirsch disease, it is due to deficiency of hepatic glycogen phosphorylase. So we can remember like this: ABCD, M to M, H to H. Out of them, the most common, overall the most common, it is going to be von Gierke, which is seen in the pediatric age group. Whereas the most common in the adults, it is going to be McArdle's. Now let me give you very quick the buzzwords how we are going to make the diagnosis of each of them. Starting with the first one, von Gierke disease. The buzzword for the von Gierke disease will be like this. It is going to occur in the pediatric age group. The patient is going to have severe hypoglycemia. Plus, there is going to be increase in the lipid levels. There is going to be hyperlipidemia, hyperuricemia. Uric acid will be more. There will be increase in the lactic acid also. So, if you are able to see that there is a pediatric age group patient, he is having severe hypoglycemia whenever in the fasting state, hyperlipidemia, hyperuricemia, hyperlactic acid is there, then they are talking about von Gierke. When it comes to Pompe disease is going to affect the muscles. In the muscles, particularly the lysosomes. So in the MCQ, if they say the glycogen is accumulating inside the lysosome, when we did the biopsy, we found the glycogen inside the lysosomes. They are talking about the Pompe disease. So the glycogen is trapped in lysosomes. If the glycogen is trapped inside the lysosome, they are talking about Pompe. Then there is the Cori disease. There is going to be increase in the branched glycogen intermediate. The branched glycogen intermediates are going to accumulate. Here also the patient is going to have hypoglycemia, but not very severe. The next one that they generally ask is the McArdle disease. That is due to deficiency of muscle glycogen phosphorylase. How we are going to make a diagnosis? It is related with the muscle. So they will say that the patient is having exercise intolerance. Whenever the patient is doing exertion, he is having severe muscle pain. He is having exercise intolerance. The moment you see this, that is going to be McArdle's. So these are the four important, out of all these glycogen storage disease, these are the important ones to be remembered. The von Gierke, in the von Gierke, the child will be there that is having the hypoglycemia with hyperlipidemia, uricemia and lactic acidosis. In the Pompe disease, the problem will be there in the muscles. The glycogen will accumulate inside the lysosome. If they mention like this, they are talking about Pompe disease, which is due to deficiency of acid maltase. If they say there is something branched glycogens are getting accumulated, that is the Cori disease, because the Cori is due to deficiency of debranching enzyme. If the debranching enzyme is not there, the branched glycogens will accumulate in the liver. That is called as the Cori disease, and that is also going to be lead to the hypoglycemia, but not very severe. The Cori disease is also called as limit dextrinosis. Then we have the McArdle disease. McArdle disease, it is due to deficiency of muscle glycogen phosphorylase, and the patient will not be able to do the exercise. Whenever he will do exercise, it will lead to severe pain, and the color of the urine will also change because the myoglobin will start coming. The skeletal myocytes will die. The myoglobin will come out and it will filter in the kidney and will go in the urine. So these are the four important out of all the glycogen storage disease. These are the most important glycogen storage disease that you must remember. Then how we are going to regulate the glycogenolysis? How we regulate the breakdown of the glycogen? We know that when we break down the glycogen, what we are going to get? We are going to get the glucose molecule. Who wants to make glucose? Is it the insulin or glucagon? Yes, it is going to be the glucagon that is going to stimulate. Insulin never wants the glucose to be made, so insulin is going to be inhibitor. There are some other stimulators are there, such as cyclic AMP, calcium, calmodulin, norepinephrine. They can also stimulate this process. Now we are going to discuss about the galactose metabolism and the fructose metabolism. When it comes to galactose, galactose is basically, it is found in the lactose. Lactose is a milk sugar. So when we consume the milk, milk contains glucose plus galactose, that is called as lactose molecule. So let's say when we are consuming the milk, the lactose molecule will reach in the intestine, and since lactose is a disaccharide, we can't absorb the disaccharide directly, so we need to break the lactose. To break the lactose, on the intestine brush borders, we have the lactase enzyme that will cut the lactose into two, glucose and galactose. Glucose and galactose both will be absorbed. Right now my concern is galactose, so I'm just focusing on galactose. The galactose will reach all the way in the liver. Then the liver is going to activate the galactose molecule by giving the energy, and will make a molecule called as galactose 1-phosphate. Then this galactose 1-phosphate can convert into UDP-galactose, and ultimately it can convert into UDP-glucose. Do you remember what is UDP-glucose? The sticky glucose molecules. And the UDP-glucose molecules, what they can make? Glycogen. And we know that how the glycogen is metabolized. So actually the galactose can convert into UDP-glucose, and from UDP-glucose we can make the glycogen. So normally it is going to happen like this, and this happens in the liver. But in this entire process, we have one very important enzyme, that is the galactose 1-phosphate uridyl transferase. This is made by a gene called as the GALT gene. This enzyme, in short, it is also written as galactose 1-phosphate uridyl transferase. So GALPUT, galactose 1-phosphate uridyl transferase, GALPUT. Now, if this enzyme is not there, we can't digest the galactose, and it is there in some newborns. For example, there is a newborn, he is not having this enzyme. What is going to happen? When the mother is going to breastfeed the child, the breast milk will contain the lactose. The lactose will reach in the intestine. It will bifurcate into two, the glucose and galactose. The glucose, galactose, they will be absorbed. The galactose particularly will reach in the liver, and then it is supposed to convert into galactose 1-phosphate, and which is supposed to convert into UDP-galactose. But because this enzyme is not there, the reaction will not happen, and these molecules are going to increase in the liver. Since this molecule is impermeable from the hepatocyte, so it is going to accumulate in the hepatocyte, and it is going to damage the hepatocyte. This child will develop jaundice. Plus, this increased amount of galactose can come in the blood, and because of that, the galactose can convert into galactitol with the help of enzyme aldose reductase. It can convert into galactitol, and this galactitol can deposit in the eye, and the child will develop oil drop cataract. It can damage the neurons also, so this child will have decrease in the IQ, and there is an increased risk of E. coli infections. This entire disease is called as classical galactosemia. So in any MCQ, if they say that the mother is breastfeeding the child and the breastfeed is not able to be digested, the child is having the vomiting after the breastfeed, means the galactose is not getting tolerated. At the same MCQ, they will say that when the glucose is given, the glucose is easily digested, no problem. But when we are giving the breast milk, the breast milk is not getting digested. It is leading to vomiting and the other problems. The jaundice is happening. They are talking about classical galactosemia, and it is due to deficiency of GALPUT, that is the galactose 1-phosphate uridyl transferase. There is one similar disease that is called as the lactose intolerance. Now, in the lactose intolerance, what happens is when we consume the milk, the lactose molecules are supposed to be digested via the enzyme lactase. But with the age, the lactase enzyme slowly will decrease. And there are some individuals, they develop the lactase deficiency in the very early phase of their life. Let's say 10 year, 15 year, 20 year, they develop the lactase deficiency. The moment they will develop lactase deficiency, they can't digest the lactose which is found in the milk. So basically they can't digest the milk. So when they are going to consume the milk, they are going to develop abdominal pain, diarrhea, flatulence, that is going to be there. This is called as lactose intolerance. So how you are going to differentiate the galactosemia from lactose intolerance? Galactosemia will occur in the newborn. Whereas the lactose intolerance, it is going to occur after a certain age, minimum that is at least 8 to 10 years. After that, this enzyme may be absent. So the lactose intolerance, it occurs later part of the life. The next is the fructose. See, when we consume the sucrose, which is also called as the cane sugar, we have an enzyme called as sucrase, which is going to bifurcate the sucrose disaccharide into two, glucose and fructose, and the glucose and the fructose will be absorbed further and they are going to reach in the liver. The fructose first will be converted into fructose 1-phosphate via the fructokinase, and then the fructose 1-phosphate will split into two with the help of aldolase B, and ultimately these intermediate will go in the glycolysis and the further metabolism will occur like glycolysis. But now what is happening here is, there are some individuals, what happens is when they consume the fructose reaches in their liver, the fructose is supposed to convert into fructose 1-phosphate and the further bifurcation is supposed to occur by aldolase B. But there are some individuals, they don't have this aldolase B enzyme. If they don't have, the fructose 1-phosphate and the fructose is going to increase. The fructose 1-phosphate, it is impermeable to the cell membrane, it can't come out and the patient will develop hepatomegaly. And the fructose 1-phosphate will be trapped inside the hepatocyte, that is going to damage the hepatocyte. And once the fructose 1-phosphate is trapped, actually the phosphate is also trapped. And phosphate is very important molecule for gluconeogenesis. So because the phosphate is trapped, this patient even can't do the gluconeogenesis also, so he will develop hypoglycemia also. This disease is called as hereditary fructose intolerance. Fructose intolerance. When they are consuming the fructose, the patient is unable to unable to digest the fructose because he is not having the enzyme aldolase B. So there are two important diseases that we have discussed. One was the galactosemia and second we have discussed is the fructose intolerance. If the breast milk is not consumed properly, that is called as galactosemia. If the fructose is not tolerated, that is called as the fructose intolerance. How we are going to remember? See here. The fructose intolerance is due to deficiency of aldolase B. Galactosemia is due to deficiency of uridyl transferase. So we have one mnemonic, FAB gut. Fructose intolerance is due to deficiency of aldolase B. Galactosemia is due to deficiency of uridyl transferase. FAB gut. Fructose intolerance is due to aldolase B deficiency. Galactosemia is due to deficiency of uridyl transferase. This is how we can remember it. Okay. Now there is something called as essential fructosuria. Essential fructosuria is due to deficiency of this enzyme, fructokinase. The patient will not have any symptom because the fructose will not metabolize, but it is not going to be trapped in the liver also. The fructose will directly come in the urine. So it will not create much problem for the patient. Only the fructose will be coming in the urine. That is called as essential fructosuria. So these patients will never come to hospital. It's just going to be accidental finding. But what is going to be important for us is going to be the hereditary fructose intolerance, which is due to deficiency of aldolase B. Right? So we have discussed the glucose metabolism, then the glycogen metabolism, then we have discussed the galactose metabolism, and lastly we have discussed the fructose metabolism.